Good morning, and thank you so much for coming to MPG Primer today. I am very excited to tell you a bit about some work we've been doing on amazing species, in my opinion. I want to tell you about our comparative genomics strategy to discover mechanisms of cellular resilience in mammals. And if you've come into the room and have just been looking at these pictures, I will acknowledge that yes, on the left, this is indeed the honey badger that don't care. The honey badger, among a bunch of other species, is one of the ones we think is really exciting with regard to cellular resilience and what we can learn from that. And today I'm going to be talking about a framework that we have used to try, or are using to try to discover those mechanisms across a bunch of different mammals. I want to start with the premise and the argument that many human diseases result from instances of compromised homeostasis. And by that, I mean there is something that happens at the organismal level that results in an organismal dysregulation of some critical physiological feature. And some examples that we may all have encountered in some contexts are temperature dysregulation, classically fever or hypothermia, glucose dysregulation in the context of diabetes, or oxygen deprivation in the context of obstructive pulmonary disease. And in all of these cases, there is a wide range of severities, but they are all united by the fact that in the case of extreme and prolonged dysregulation, there can be really aberrant consequences for cells that can result in protein deterioration and in some severe and prolonged instances, cell death. Many of the treatments that we have for these disease states are aimed at restoring homeostasis and normal levels at the organismal level. So in the context of temperature dysregulation, the idea might be to take someone who's hypothermic and warm them up, or take someone who has a fever and cool them down. For glucose dysregulation, insulin could be used to restore typical blood glucose levels. And in the case of oxygen deprivation, to pursue interventions that will restore an individual to a normal level of oxygen in which their cells can reside safely. I think that one of the things to keep in mind about this is that the reason we have to do all these things and the reason these dysregulations are bad for human cells is that under normal conditions, human cells reside in a pretty reliably good environment. And so one of the sort of examples that I think is useful is to think about if you are hiking in the cold, your hands might be cold, you might be thinking, gee, I'd really like a cup of tea, but your body temperature will stay right around 37°C in spite of being in an environment that's considerably colder than that. Even more amazing, I think, is that that is a bidirectional tolerance. So if you happen to like hiking when it's hot out or in places where you have to tolerate heat to get amazing views in the desert or wherever, you will again find that your body is staying at about 37, even though the external conditions might be much warmer than that. And I think this is really interesting because it, to me, suggests or indicates that humans are really tightly controlling our internal conditions. And, on the one hand, you might think that this would be an easy problem to solve, but it turns out that to do this reliably at population scale has for a long while been quite difficult. And many of us are wearing on our wrists part of the recent solution to that problem, which is to wear various wearable devices that are collecting temperatures over a long interval. And Houseman et al. Back in 2018 used these as a data source to try to begin to understand, well, how much does human temperature variation really, how much is it a thing, basically? And so this is an interesting paper that looks at a bunch of different individuals, but the overall conclusion is that if you take all those individuals, you take means, and there's some interesting variation, but you take means, you'll find that, yes, indeed, there is variation in human temperature over the course of a day, but that y-axis is pretty tiny. And so you might find if you were to wake up in the middle of the night at 4 a.m. and stick a thermometer in your mouth, you'd be at around 36.2 on average. You'd be a bit warmer at 5 p.m. if you did the same thing. So there is some diurnal variation. But again, this is a pretty small, like 1 degree level range. But there are limits to this. And I was really struck in a couple of years ago to see a New York Times feature about exactly this. And, sort of we have evolved in sort of through human history in environments that have certain limits on what temperatures are typically attained in the environments that we live in. And this is really striking to me that in some cases, there are now parts of the world where it's not all that unusual for temperatures in places that humans live to get up to 100, well into the 100s, basically. And even though, again, as I said earlier, we are usually working on the systemic level to keep our internal conditions thermally pretty constant, those conditions seem, in some cases at least, to be sufficient to overwhelm us. And so, as the New York Times phrased it, the heat of the day can give you a fever. So basically, not only is sort of excessive body temperature a disease state, but it's now actually becoming something that is not all that implausible as something that our environments can impose on us as a stressor. And so what does this mean? Well, I want to think about what this means in terms of the following statement, which is that humans are pretty weird. In fact, as I was telling you earlier, if you're hiking in the cold or if you're hiking in the heat, your body is working pretty hard to keep you at a constant temperature. If you were a snake, this would be less likely to be true. In fact, as we all, I'm sure, encountered way back in elementary or middle school biology, snakes are like reptiles in general, very much conforming to the ambient temperatures. And so, broadly speaking, if you crank up the temperature that your snake, if you happen to have a pet snake, is living in, the snake's temperature will go up as well. And so snakes have behavioral strategies of like seeking out sunspots in order to stay warm. And again, that's because they're not really working very hard at all to keep their internal conditions constant. But even mammals, even among mammals, we are weird, I would argue. And so one of my favorite animals, which I sort of anticipated in the very first slide with a few photos, is the dromedary camel. And I love this picture because the camel looks so unaffected by whatever is going on, just chilling there. It turns out that if you are a dromedary camel, you have some fluctuation in temperature that is, this has been known since the mid-50s, that is very related to the temperature of your environment. And so, you know, if you think back to the snake, which was like basically the snake is the temperature of the environment, that's not quite as extreme for the camel. And so if the camel is in a freezing environment, it's not like the camel will freeze, but you do see some variation in terms of the body temperature that's in excess, like 3.5 degrees, which is in excess of the diurnal variation I mentioned earlier for humans, where the camel is letting its body temperature vary a bit. And the sort of canonical thinking about this, at least, is that this has evolved as a strategy for water conservation. And so, you know, if you're human, one of the ways you deal with keeping or you achieve a constant internal temperature in a hot environment is to sweat. That's all well and good if you have access to water. It's kind of a terrible strategy if you don't have access to water. And so it seems like sort of an evolutionary trade-off where the camel is tolerating internal variation in temperature as a way, very hand wavy, but it seems to preserve water. And it turns out camels aren't alone either. If you are familiar with the process of hibernation, you're also aware that there are many species that kind of allow the temperature perturbations in the opposite direction. So I featured two of my favorite groups here, the bats and a 13-line ground squirrel, both of which have really, really substantial temperature variation over some cycle during the winter. And this too seems to be a trade-off. And so if you hang out in the evening in the summer around here, you'll see, and we all benefit from all those bats that are hanging out and eating mosquitoes. But it turns out that come frost and come the cold weather around here, there aren't mosquitoes available. And so this is not the species we have around here in particular, but the ability to kind of hang out in the winter and not need food by letting your metabolic processes go into a more depressed state is actually a really good way to deal with a lack of food. One thing that I'm not going to talk about today that I think is kind of interesting is the very different patterns, and this is just an aside, but it's something we want to know more about, of the temperature variation in different species. And so hibernation isn't necessarily getting cold and staying cold. It's maybe, as more on the squirrel end here, having these intervals during which you're fluctuating a lot. But when you are really cold, you're saving all of the resources that you might otherwise be investing in staying really warm. So in light of this, in light of the camel getting really hot, and in light of the hibernators getting impressively cold, I would argue that mammals exist along sort of a continuum of strategies for coping with environmental variation. And so as I was mentioning before, humans are kind of weird. On the left, I would say that we have really strict homeostasis. We work really hard to keep things internally the same. And we do that at the expense of water. We do that at the expense of needing to spend more energy to preserve our rather high body temperature, even when it's cold out. But if you happen to be a camel or a squirrel or a bat, there are other resources that are of greater value, in part just because they're more limited in the environments that those organisms live in, either because of their dietary habits or because of water limitation in the case of camel. And I would call these species that are more on the end of flexible homeostasis, where they are tolerating internal conditions that are variable. Now, where does this place the expense that those organisms face? And I would argue that it's really sort of, again, a continuum between organismal mitigation and cellular mitigation. So humans are working hard on systemic level, but if you happen to be a camel or a bat or a squirrel, it's actually your cells that are directly encountering those variable conditions. And so this really, I thought, was really an intriguing thing. And in fact, just as an aside, that graph I showed a couple slides ago of the camel having variation over the course of the day was something I actually encountered in an undergrad comparative physiology class. And I thought that was so cool because it totally violated what I thought I knew about mammals being very human-like. And again, I think this comes to sort of the downsides of having a human-centered view, which is that you think we're normal, but we're weird mammals. And if you start thinking about the diversity in this continuum, you suddenly open yourself up to the possibility of thinking, if there are species where it's the cells that mitigate the stressor, then that allows us to think about this whole other set of questions, which is basically how do mammalian cells buffer or cope with stressors? And this opens up another set of more specific questions. And these are the three that we've tried to use to frame our project right now, although I think, as you'll see later in the talk, I'm beginning to realize that these are not so readily addressable as I had hoped, and that's where I would love to have your input on what to do next, because we are uncertain about where to go. So I think there are, this can be broken oversimply into a bunch of ways. One of them is which responses are universal across species. That is, are there certain things that you do regardless of whether you're a flexible species like a camel or a strict species like a human? Are there responses that are unique to flexible species? That is, are there things that are that the camel can do that we can't? And finally, which of those responses drive all the others? Like, is there one that sort of precipitates in any sort of way, genomically or otherwise, the set of subsequent responses? And I will say that, I will admit that my interest in this comes from a very basic biology perspective. I just think it's really cool that cells can do that, and I want to know why. But since we have tried to pitch this to NIH and currently have funding from NSF, we have challenged ourselves to think about what this might mean in the context of what is this, what might be the insights for humans. And I really think one way to think about it is that instead or complementary to ideally, strategies that are meant to sort of improve conditions at the systemic level for disease states of temperature, glucose, or oxygen dysregulation, a complementary strategy, if we can learn from other mammals about what their cells do that is special and awesome, we might actually find ways to intervene to promote cellular resilience. And so, if you can't necessarily totally normalize blood glucose, or you can't normalize temperature, are there other things that we can do that will protect cells even in a more variable environment than they typically live in? And this comes from, again, an evolutionary perspective that we may not be drawing on these mechanisms as part of the typical lives of our cells, because we are generally doing a good job of keeping them in pretty constant conditions. But in these disease states, is there something we could do that can be informed by our shared evolutionary history with all these other mammals? To approach this, we started thinking about what we might learn from sort of really classical ecology experiments. This is, I'm sure, I've got a typo there, sorry, but when you take a bunch of species or a bunch of individuals and you culture them under the same conditions, there's, as I'm sure many of us have encountered before, a common garden approach where you basically do the same thing to everyone and see how they differ, and figure out what are the intrinsic responses of those species or individuals that allow them to cope that allow them to produce different phenotypes in the same environment. And we have been able to take this approach thanks in large measure to some amazing resources at the San Diego Zoo Global Frozen Zoo, which has stored dermal fibroblasts from thousands of species. And this is the key part, including multiple individuals per species. And so there's this amazing room, which I would wager to say has more viable genomic and genetic diversity in it than anywhere in the whole world. And it looks kind of unremarkable in what it is. This is definitely not as exciting to visit in the sense of, oh my gosh, I'm going to observe animal behavior as a zoo, but there's a lot going on here. And we think that this is especially exciting for a number of reasons, or has been especially exciting to us for a number of reasons. One of them, as I've indicated here, is that rather than having one sample, so a cell sample from one individual of each species, the zoo has had the forethought over many, many, many decades now to sample cells from multiple individuals per species. And for anyone who's ever designed any sort of experiment, you of course know that this is really essential, because what we don't want is, we don't want to say, hey, look, this camel is doing this thing, human is not, this is a camel thing. We of course want to know about intraspecific as well as interspecific diversity and responses. And so we've been really fortunate that there are many species, mammals and otherwise, for which the frozen zoo has stored cells from multiple individuals. I want to give you a sense, just starting here, of what the set of species is that we have. So as with any experiment, this was in part, this list, the composition of this list was in part driven by, we think these are the most exciting possible study species, and in part by the practicalities of what's available and what can be cultured. We have, as you'll see here, the dromedary camel. We also have the Bactrian camel, which, even though it resides in much colder conditions, seems also to have the ability to let its temperature vary a bit with the environment. We have a bat and the squirrel as at least two hibernators. We have a couple of species that we think are really interesting in other ways. So the gelada, for example, is a really high altitude primate that lives under oxygen limited conditions. And I'll come in a second to why we think that's important as well. And the Egyptian fruit bat survives basically on a fruit and a fruit diet, which would obviously not be a super sustainable strategy glucose regulation wise for a human. And so we think it's interesting in that way as well. This list is, there's been some variation in this list, but we are happy that we have as many species as we do, and simultaneously frustrated that we don't have more, because statistical power is always a challenge with any project of this sort. And so, as I'll come to a bit later in the talk, we are currently trying to figure out what we can do with the data as we have them, and what the limitations are given power.
Diane?
Yes.
Yeah. I might have missed this, but have you thought about introducing or adding evolutionary distance among these species and making sure you're covering lots of millions of years of evolution?
That is a great question. And there are two answers that are sort of, but not really opposed. One of them is we've definitely thought, and maybe more accurately, worried about it. And I'll come later to a more explicit discussion of why we think that's really important. We would love to have, the ideal scenario, as I think your question is alluding to, is to have multiple species that have the same types of resilience that are deeply diverged from one another, right? Like, if you want to know what makes you able to cope with really variable temperature conditions, you want to be able to disambiguate, hey, I'm a camel, this is the thing I do, from, hey, look, this is an essential type of response for a mammal to be able to cope with variable conditions. In an ideal world, we would have much greater coverage of that simply by virtue of having more species. We did, to the limited extent possible, try to build that into our design with constraints that we are not super happy about. So with some specific examples, as I was saying, we think that the camels have some really interesting thermal stuff going on, but they're close relatives. The oxygen limitation stuff that I'll come to in a second, we have a little bit better phylogenetic spread, basically, because the seal and the gelada are on quite different branches of the tree, and both encounter that as part of their normal environment. So in summary, we did think about it. We haven't accommodated it as comprehensively as we would like in an ideally designed experiment. But ask me again in some number of years, and I hope I'll have a different answer. And again, just to this kind of was the question you were anticipating, but it turns out that for the species we've decided to include, a number of them have at least one and in some cases multiple types of flexibility. And so, as I was saying, the hibernators and both camels seem to be thermally flexible. The gelada and honey badger and Egyptian fruit bat all seem to have some glucose flexibility. We've got 2 diving mammals or 3 diving mammals rather, and then also the gelada as organisms that are tolerant of hypoxia. And so hopefully that to some degree gets at this, and I say hopefully more emphatically than I would have a couple of years ago. I thought we could do more with it than we can. And so we've kind of switched from, we're going to find all the things, to now we're going to try to accommodate the limitations of our data set, do a proof of principle, and hopefully convince someone to give us more money to do truly all of the things. Okay. Are there any other questions or comments or suggestions before I go on? Okay. Cool, great. And just to give us a sense of what we mean by the variable conditions, I've been talking mostly about temperature, but we wanted to do sort of a truly comprehensive framework. And so in addition to temperature flexibility, we considered glucose and oxygen flexibility. The temperature conditions are selected with constraint. And again, this is another one of the kind of comes back to your question, Nicole, about the practical versus ideal aspects of experimental design. So we decided to choose as baseline for all of our stressors the conditions that are typical for culture of human cells in the lab. And so 37 being human body temperature, that's a reasonable starting point, we think. For the low temperature, in an ideal world, we actually would have been able to go lower because, as I showed you before, hibernators get really, really low. And so 32 isn't that cold if you're one of many species of hibernators. The downside is that we wanted, or the constraint is that we wanted to create a truly comprehensive data set where we had data from every individual of every species at every condition. And so it turns out that if it gets too cold, some of these cells just don't grow. And so this is another thing that I think is sort of one of the trade-offs between having a comprehensive data set and having a comprehensive set of species, because we can't figure out how some cells do it much lower than 32 because they just don't do anything because they don't survive. And so that was one constraint. For glucose, we tried to model by our choice of conditions the conditions that might be pretty severe hypoxia. Oh, sorry, not hypoxia. Low glucose and high glucose that might correspond to a hypoglycemic event versus a hyperglycemic event. And then for oxygen, rather than exposing species to high oxygen conditions, we actually decided to do short and long exposure to hypoxia to sort of model what a short stint versus a long stint for a mammalian diver might be. Again, this is a sort of a less comprehensive set of conditions, and we'd really love to have a more continuous distribution of states across, or of conditions across these extremes, but we started here. And then for data types, we decided we wanted to learn about as many different features of cellular response as possible. And so our collaborators on this project who are focused on cell physiology have a bunch of different ways, I'll mention a couple of those in a second, to figure out how are the cells doing under these conditions. And so for every individual of every species under every condition, we asked some basic things that we think are enabling us to get at the question of cellular health, for lack of a better word. We also asked about gene expression, which is the analysis that we're furthest along in, but we have still reached an impasse with that and would be grateful for your input. We have some chromatin accessibility data that we're very much at the entry stages of analyzing. And that same goes for some cell morphology data where we're trying to figure out really basic, but I think potentially quite informative things about how does cell shape change? How does the mitochondrial shape and distribution spatially change under these different conditions? So again, our collaborators, Alison Hindle and Carla Medelaire, have been focused on the cellular physiology aspect of this project. And we really, for the cell physiology data, started out just by sort of doing a proof of principle. And so our prediction was that if a species is flexible in its temperature range tolerance, then it should be able to better cope with variable temperatures in a way that's observable at the cellular level than one that, like a human, keeps things really, really tightly controlled internally. And I was very anxious to get the results of these experiments, in part because if it turned out that we didn't see any variation between the strict and flexible species here, we'd kind of be in a bad situation, right? The whole sort of premise of the project is predicated on the idea that cellular responses to variable conditions in some way capture or recapitulate organismal phenotype. And if we couldn't observe that in culture, we would have been very sad. So I am very relieved by this graph, and I think maybe the food is arriving. So maybe this is a good time to pause. Is that good? So I will leave you to think about this for a second while you grab some coffee and food and then finish up in the next couple minutes. So great. Yes, please. Yeah.
So when infants have an initial hypoxia event around the time of birth, we actually cool their core temperature to 32 degrees to actually prevent cellular injury. So after hypoxia, the cooling actually lowers metabolism enough to prevent secondary cell death. So I just find that really interesting to think about in the context that our organisms are, again, what you were saying about cellular resilience versus organismal resilience, right?
Yes.
That a whole organism can be 32 degrees as a human, but that our cells can't be.
Yeah.
So I just found that interesting.
That is really interesting. And it also makes me wonder about another thing we haven't yet gotten to, which is the combinatorial thing. So it sounds like the scenario you're mentioning is sort of the effect of cooling in the context of hypoxia, and that maybe something that this, I mean, not exactly misrepresents, but doesn't get at, right? Because it's only looking at cooling in the absence of hypoxia, where I'm sure there are trade-offs at all. Thank you for mentioning that. Yeah.
But it also makes me wonder, right? Like many of these animals that might have a shorter lifespan might have less genetic pressure for some later onset disorders or things relative to those that would prevent reproduction.
Yeah.
So I wonder if just how the kind of life cycle is integrated into the thoughts of which genetic pressures would be experienced by each species.
Yeah, that's a really good point. I think that the short answer is we don't have enough data to include that as a covariate as comprehensively as we would like. The slightly longer answer is that I suspect that if we did have a more comprehensive species set, we could start to get at questions about whether the stressors are routine or episodic. So for example, if you're a hibernator, you have to be able to deal with cold every year or you're not going to make it to spring. Whereas if you're a species that lives in an environment where it's occasionally really hot, that would be more of a selective sweep on occasion than a constant pressure. And I would think that that would impose some really different outcomes that you might be able to parse apart if you had enough species that are in those different scenarios. Basically, we cannot get at that, but we've thought about that a bit in terms of the hypoxia things, and that's why we've thought about the time of exposure. But yes, I think that's a really good question that we can't yet address at all. So.
And then my last question, which is like a 9th grade biology question, is it seems like you're looking for episodes of convergent evolution, right?
Yeah.
Where like very different species have come to the same like nodal pathway as their solution to a stressor. What if you looked for divergent evolution, like two species that because of a change in ecosystem specialization, no longer, either one continues to have the stressor and the other one no longer does?
Yeah.
And if you found multiple of those pairings, that could also show you what is shared and changed.
Yes, definitely. Again, more species would be lovely for addressing that. I think what perhaps you're talking about is in an aspect of what you're alluding to is that there is the potential for a relief of selective pressure, right? And so there are some sort of parallel approaches. They're asking for specific genes that have had accelerated evolution or stronger purifying selection, for example, that might be more directly able to get at the when did this happen and how over what time scale is it continuing to happen. Again, another set of amazing questions that we have worried about and not been able to address in this context. So yeah, thank you so much. Thank you.
So please, everyone, feel free to grab breakfast. We really apologize for the lateness. Diane, if I could ask you a question that came from an anonymous attendee online, again, about more species. They say, have you considered extending your study to avian species? To your examples, their internal temperatures, etc., is a bit more constrained than reptiles, but not so well as mammalian species. It might give clues about mechanisms conserved over much longer evolutionary timescales.
Can you read that second part again? I just didn't hear one of the keywords.
Oh, I think they're just asking about adding avian, and then gave some really interesting biology about how that they can constrain their temperatures a little bit intermediate between reptiles and mammals.
Yes, and which of course is perhaps not surprising given the tree structure as well. As with my maybe unsatisfying answers to the other questions, yes, we have considered it. No, we haven't gotten to it yet. But I think there are some really interesting things about birds, certainly, that I might wildly guess that there are some features about how birds respond that will be shared with mammals and others that maybe are unique, having to do in part with their evolutionary distance and also with the extent to which they tightly control things. So it's a wonderful question and I have no answer. So maybe I'll take like 2 more minutes of pause, 3 more minutes of pause, and then get back. Okay.
Alert your pediatrician.
I think I may throw that question to Sarah. Can you ask it again? I'm sorry.
Oh, just because, like, if you have a kid that's got a, infection of some description, and they actually can have temperatures of 104, but pediatrician didn't get upset about that,
Yeah. It's in some ways, it's the rate of rise or the change in temperature that's more important for things like risk of febrile seizures. So you're right that, the 104 in and of itself actually makes it more likely to be viral, which is part of why people just don't have therapies to offer other than antipyretics. But in terms of like clinical complications of a very high fever, it's really like 106 or higher where you start to get like cellular damage, but it's the change in temperature, so either fast up or fast off, that's associated with seizures. So those are the things we worry about. We're telling you,
but the temperature is 104.
Yeah, and right, and they look miserable, right? There's organismal compromise, but not cellular compromise yet.
Whereas there would be for an adult at that temperature. Is that right, or is that just less commonly a state attained by adults at all?
That's a really good question. I think of it as being something that's more recoverable as long as it's not sustained, right? Like I feel like humans very rarely really sustain a temperature that high. It's usually on that order of hours.
Yep.
But this is all like phenomenologic recollection from clinical training. So there might be like much more detailed information out there.
Yeah, cool. Thank you.
Any other questions while the less people get their breakfast?
Yes, first of all, this is super interesting. Like this comparative biology perspective is really fascinating. But I was wondering, just like from a different understanding, because these are all fibroblasts. Like, would you expect something different with hepatocytes or cardiomyocytes, or like is there? Yeah.
It's a really great question, and I think you're probably detecting a theme here, which is that I have a idealized response and a pragmatic response. And I'll start with the idealized response, which is that in a way it might be nice to be able to, or especially nice to be able to get at some cell that is not so directly exposed to the environment that it is, closer to what the whole organism and the organism's internal features are experiencing under these conditions. So I would love to be able to do exactly the same experiments in parallel with one of these other things, because in addition to it, there being an argument that would be a better cell type, whatever, it also would be really interesting to compare to see whether this is this a camel thing, or is this a camel cell exposed to the external conditions thing. So my response there is yes, definitely. My response about dermal fibroblasts in the pragmatic sense is that one of the things that I really appreciate among many about the San Diego Frozen Zoo is that they have been dedicated to minimally invasive sampling. And so all of these cells were collected in the context of routine veterinary care. And so, to collect from an internal tissue would definitely require invasive and maybe immediately post-mortem collection. But the reason, at the moment at least, that cells are available from such a great number and diversity of species is that this is an accessible cell type. There's also, I think, the argument that dermal fibroblasts being outside, basically, are going to be more exposed to the environment. And so we might expect that they are typically experiencing these fluctuations more directly. And so we think, hand wavy, we think that maybe we would have greater opportunity to observe some of these things. And so it felt to us like a reasonable order of operations to start with the cell type likely to yield the greatest effects, and then move later to exactly the ones you're mentioning when those things become available. But I would love to know how many of these things are cell type specific versus species intrinsic things. And I do not know. And we won't know for a while, I think.
All right, thank you.
That's a really good question.
That sounds great. It sounds like most people have their lunch, and maybe we can wrap up in the next 15 minutes.
Oh, yeah.
Do you think you could finish
that
by 25?
Sounds great. Yeah, I think I was aiming for 9:20, so I think we're good. Yeah.
Thank you.
So I've had this slide up here for a while. Basically, I just wanted to make the point that oxygen consumption rate, our collaborators who work on cellular physiology use that as a metric of how the cells are doing under a given set of conditions, because it is a reflection mitochondrial respiration rate. And we were encouraged, although the data are very noisy, to find that in many cases, and not all, obviously, but in many cases, the strict species have more of a change between their oxygen consumption rate at baseline, which here is represented as the 0. So that's no change from 37 when you expose them either to 32 or 41. Than do the flexible species. And so each of the symbols here is an individual within that species. And so this comes to why we thought it was important to have samples for multiple individuals per species, because we can attempt, with the limitations of N, to sort of disentangle a species feature from an anomalous feature of a particular individual. Obviously, there are some exceptions here, and we would love to have more individuals to know what to make of them. But for example, humans don't seem to respond as badly to 32. And maybe this comes to what Sarah was mentioning earlier about the potentially beneficial therapeutic effects of that. But we just don't know. But anyway, as an initial pass, we were enthused to see that the strict species seem to respond with greater change in oxygen consumption rate to thermal perturbation than do the flexible species. One other point I wanted to make is that, as I've mentioned earlier, the camel and the squirrel, even though they're both okay, it seems, at organismal level with temperature variation, they're okay with temperature variation in different directions in their normal lives. And so the camel typically gets really hot or comparatively warm. The squirrel gets cool during hibernation, and yet both of those species are okay, it seems, for oxygen consumption rate, both at 32 and at 41. And so one of the emerging themes that I wouldn't go so far as to say is a pattern yet, but we are intrigued by, is the possibility that there are some components of thermal tolerance that are non-directional, meaning that maybe there are some things you evolve that allow you to deal with temperature perturbation that are not specific to getting warm or getting cool, but just are about being able to deal with greater temperature variation. Another aspect of the data that is very much in its infancy, but as I was mentioning earlier, our colleagues who work on cell physiology are also taking using microscopy to examine cellular impacts at various temperatures. The white rhino, as you'll recall from this previous slide, is one of the species that is a strict thermoregulator, we think, and seems to do especially badly under both hot and cold conditions. So it has these significant changes in its oxygen consumption rate. And we also noticed, and again, these data are very preliminary. I'm sorry that there isn't a species to compare them to just yet, but it seems that the rhino cells have a lot of changes in cell morphology when you either cool, or sorry, in this case, when you warm them. And so we're interested in asking, as the data begin to emerge, which they are doing now, whether this is a general property of, for example, species that have aberrant changes in oxygen consumption rate under thermal perturbation. I want to end with talking a little bit about what we've done so far with the RNA sequencing data and what we think is a major constraint right now and what we can do with those data, and ideally ask for your help in coping with that problem. So when I first started thinking about this project and this problem, I was like, oh, this is great. We're going to take all these species, expose them to these conditions. We'll find things that everyone does and that just the strict or flexible species do when they're exposed to thermal perturbation, for example. As Nicole was alluding to already, there's a big problem with that because we don't just want to know, hey, what do strict species do or what do flexible species do that strict don't? We really want to know, are there things that are surprising given the phylogeny? And so I had not thought adequately about that constraint before we started this. And so right now we are able to say things like, there are universal responses among all study species. And by this, I mean, if you crank up the temperature to 41, there are certain pathways that are enriched among the genes that have a significant fold change when you use all 5 of those individuals per species that every single individual does. And so some of those have to do with cholesterol biosynthesis, some cell membrane stuff there, maybe not totally surprising in light of a response to higher temperature. There are some things having to do with protein folding, again, not surprising given some of the things about protein denaturation. So that was all good. These are things we can say everyone that we've studied so far does. We can also start to say things about what our star flexible high temperature species, the dromedary camel, does in terms of regulating pathways or changing the expression of genes in pathways that other species don't do or that strict species don't do. And those have to do with cell cycle proliferation, protein homeostasis, DNA repair, protection against oxidative stress, and homeostasis of mitochondrial metabolism. This is maybe not surprising. These are all things that might go badly when it's high temperature. I was initially really excited about this, but then I started worrying that this is just, again, still very descriptive, right? We can say that these are things that camel does, but we can't really say whether they are, well, we can say that in our study species we're unique. We can't say that they're universally unique. And that things get even worse when we think about the constraints of our tree. When we think about trying to find trait-relevant convergent changes in gene expression, which we really think is a complex challenge, and the asterisk there is to highlight that this is one we have yet to solve. And I've been talking with a bunch of people who work on phylogenies and evolution of traits, and I have been really struck that we're kind of maybe as a field converging on this as an open question that doesn't yet have a really satisfying and statistically appealing answer. So I just want to take us through this example. And again, this comes back to something that Nicole was mentioning a couple of minutes ago. So say that we observe that our 2 camel species, both of which, as far as we know, are thermally flexible, have some gene that is either up or down regulated significantly in response to temperature. I personally would find that exciting. We can say this is the thing that camels do when it's really hot. But there's a problem in that this also maybe isn't totally surprising because those camels are closely related to one another. And so those aren't really independent observations. They're a thing we know about what camels do, but we can't say that this has evolved independently or this is a response that is sort of universally present among species that can tolerate high heat. At least in the relative sense, I would be more excited if we were to find some gene whose expression changed significantly, not just in the camels, but also in the hibernators for 2 reasons. One of them is that camels are heat tolerant. Hibernators seem to be more tolerant of lower temperatures. And there's a much greater branch length between our camels and our hibernators. And so that would leave me comparatively confident, also very hand-wavy, that this is a sort of a convergent trait that exist independently on these lineages and isn't just sort of a byproduct, if you will, of their recent evolutionary relationship. So my colleague Lucas Moreira and I have been scouring the literature to try to find about whether there are ways to that exist already to try to understand and put p-values on basically a sharing of responses across species with a given phenotype that is impressive given their phylogeny. And so one of the methods that has begun to get at some of this is the Eve method, which the expression variance and evolution model by Rolfs, introduced by Rolfs and then elaborated more recently in a bunch of other papers, that basically assumes that gene regulation, as shown here on the left, evolves randomly across a tree. And so the intuition here is that if a branch length between two species is longer, then you'd expect them to be more diverged than the branch length in expression change might be between two closely related species. And so this is like a classic random walk. And so the idea here is that you just simulate along the tree, and what you're looking for is cases where the expression change on a given branch is excessive given the length of that branch, which would suggest, yep, there's something really, really meaningful going on here in terms of the change in expression. This model is also able to look at a bunch of different species and their fold change in response to something and ask about whether there's evidence that there are two different optimal levels of expression. And so they call this the theta test. And so basically you can ask, is there evidence that there are that maybe high expression is selected in one lineage and low expression is selected in another lineage. This was really exciting to us at first, and indeed it's an awesome method, it's super cool. The problem is that there is not yet, as far as we can tell, a way to specifically look for cases of convergence. And by this I mean cases where you, again, as you might think about in this example, are there things where, in spite of the long branch link comparatively between the squirrel and the camels, there are things that are more similar in their expression change than you'd expect given that lineage, given that branch link. And so I will just stop by saying we've been running Eve, which again we think is an exciting start, but it leaves us kind of with this problem. And so we'll see that there is evidence, for example, for this gene, which I'm just using as an example, for two different optimum expression levels. That is, there are significant differences in the expression of this gene for some lineages as compared to the other. However, what we can't do is to say that the flexible species, so here the squirrel, the bat, and the two camels, are more similar than you'd expect given their branch length. And so we've been trying to figure out, are there Ford simulation ways to get at this? Are there other things that we haven't thought about? In talking with a bunch of people, as I was saying recently, about sort of a phylogenetic approach to this seems like a problem that the field is converging on in terms of, no pun intended, but the field is converging on in terms of looking for genes whose expression is more similar between diverged lineages that you can annotate a priori than you'd expect. I don't know what to do with this. I will close on the unsatisfying note of saying that we are still perplexed by this. And for me, it's kind of a sad note in what I think is otherwise a promising project where we can right now say, hey, look, these genes change in these species, but I cannot tell you whether that is surprising given their phylogeny. And I hope that not too long from now, we collectively will come to an answer and I will be able to give you some greater certainty on that. And then let's see. So finally, this kind of comes to what I was just saying a second ago, but we want to identify convergent changes in gene expression, convergent changes in chromatin accessibility, and then in some work I haven't had a chance to talk about today, convergent changes in RNA splicing in the response to temperature and other changes. And what we really want to do, I think, is develop methods to get at these things. So with that, and I think my timing is okay. Unsurprisingly, given the diversity of data types that have gone into this project, we have a whole bunch of collaborators working on a whole bunch of things. So Lucas, who is up here on the left, who was at Broad and is now at Colossal, has done all of the RNA expression work and has been trying to see whether there's a way we can push the Eve model to get at this question of convergence. Carla and Allison here have been doing all of the cell culture work, without which none of this would be possible. Eleanor and Lucas and I have been focused on the genomics. Jorge and Christy and Valerie have been working on some mathematical modeling to try to get at the question of genomic drivers of these responses. Daniel is starting to work on the chromatin accessibility data. And then I also wanted to give a shout out to the Bird Summer Scholars Program, through which we had the opportunity to work with 2 high school students this summer who really started doing some RNA splicing work with our data that this was news to me, but we just had RNA expression data, and with those and combination with a splicing program called RMATS, we were able to infer instances of differential splicing. And we think that there may be a lot of instances of differential splicing thanks to Jordan and Eric's work. Jordan and Eric's work. There may be many cases of differential splicing that are also responses to heat that we would miss if we were just asking about changes in expression. I will end there and say, I'm sorry that I didn't have more conclusive things to say, but this is where we are in this project, and I hope we will be further along in not too long, and I would be grateful for any questions and suggestions.